Okay, so now we have Manolis. Manolis is up, and Manolis is a great expert in regulatory genomics and single cell, and he's going to give a kind of guest lecture here just for a bit and tell us what he's doing with that.
Hello, everyone. So thank you so much for having me over. It's a pleasure to be here. I'll give a broader overview of the field rather than only focus on what my lab is working on. And I put a copy of my slides in Dropbox, so hopefully all of you guys will have access to that. Let me find where that is. Oh, for some reason.
We'll post all that stuff, Manolis. We'll post it all up for people.
Okay, so there you go. I'm putting a link to the chat. Great. So what I'd like to tell you about today is how we use deep learning in the context of predicting TF binding, predicting single cell profiles, and more generally, deep learning methods in genomics. So this should be a review of the basic foundations of gene regulation. So as you know, we have one genome. Every one of us has one genome. And that results in this incredible diversity of cell types in our brain, many different blood cell types, in our heart. Even just a single snippet of your skin has all of these intricate relationships and webs and networks of different cell types interacting with each other. How is this possible? This is possible through epigenomics, through the memory of every cell type of what is its identity. And the way that this memory is encoded is by DNA wrapping around these balls that we call nucleosomes, where about 150 nucleotides of DNA and a 50 nucleotide spacer is between every one of those. And those have different colors. And the colors are established through post-translational modifications of these long keystone tails that make up nucleosomes. And that allows us to now distinguish, say, the orange enhancers from the red promoters, from the green transcribed regions, from the gray repressed regions, and so on and so forth, all of which is possible through specific combinations of histone modification marks, DNA accessibility, and DNA methylation. So there's been a lot of methods for learning combinations of those histone modifications and those epigenomic modifications that allow us to now predict the state, quote unquote, of chromatin at every location. That allows us to distinguish different classes of gene regulatory regions, such as enhancers that act at a distance, promoters that act proximally, transcribed regions where polymerase goes through to produce protein coding genes and non-coding genes, and that are usually marked for non-reinitiation. So these are actually marks that very often prevent restarting there, and then repressed regions, and so on and so forth. And the cool part, the cool part about all this is that it's all encoded computationally. So we can basically discover the words that make up these gene regulatory regions. So we can discover motifs, which are short DNA sequence patterns that are responsible for recruiting proteins that recognize these elements and that establish the chromatin environment of an enhancer region, of a promoter region, of transcribed regions, and so on and so forth. And these motifs are everywhere. Now, what's really cool is that you can take a cell from your skin and basically make another you by reprogramming that cell, stripping its epigenomic memory, that it's a skin cell, and then reverting it to a pluripotent state, which you can then differentiate into any cell type that you want to have more of, or into another copy of yourself, to have spare parts and so on and so forth. I mean, of course, with a little bit of science fiction ahead, but all of the parts for all of this work are slowly coming together. So what we are going to focus on today is what are the building blocks of genomes and how do we use comparative genomics, epigenomics, deep learning, motif discovery to discover those systematically. So the foundations of all this is that transcription factors bind DNA, not by opening up the bases and then sort of binding by complementarity, the way that RNA would bind DNA, or that CRISPR would recognize different parts for targeting its cutting of DNA. But instead, they're feeling the DNA from the outside based on specific DNA residues that are on the base pairs and that are interacting with the transcription factors. So the transcription factors come in many shapes and forms, and they interact, of course, both with the phosphate backbone as well as with the bases themselves. And they can really interact with any nucleotide, with any molecule on these bases, any atom on these bases. That basically means that the corresponding sequence patterns that these transcription factors bind can be sometimes degenerate. In some cases, it recognizes 2 or 3 atoms that really uniquely define a T. In other cases, uniquely define a C, uniquely define an A, and so on and so forth. But in some cases, it recognizes only some atoms that don't really distinguish an A or a G. So you will end up with an ambiguity at that location. And it doesn't really matter if you have an A or a G or a C or a T at that position, you will still be able to bind equally strongly. So I want to sort of get a feel for the class, for who's following me here. And I don't know how I can do that. So give me a thumbs up in your reactions down there if you're following. I don't even know if reactions are possible. Oh, good. I see a few thumbs up. Good. Awesome. Lots of thumbs up. Any questions so far? So basically, this is just the most foundational building blocks of regulatory genomics. So number one, motifs are everywhere. They're there in enhancer regions, in promoter regions, in splicing, in post-transcriptional regulation. Number two, motifs are defined by the interactions between transcription factors and DNA, at least for pre-transcriptional regulation, and in some cases by microRNAs binding 3' UTRs, or anywhere in the transcript, or splicing factors binding within either exons or introns, and we're going to talk about all of the above. So having this information, we can now distinguish different motifs from each other and distinguish different instances of motifs from each other. So this is the core building block of regulatory genomics, a position weight matrix, also known as a position-specific score matrix. What is that? It basically tells you that when I line up all of the instances of a particular regulatory motif, I'm going to find A and G most frequently, and here nearly always a T, and so on and so forth. Notice that in some cases, I don't find that exact letter, and the motif matches don't need to be exact. They just need to recruit the transcription factor. So the question is, first of all, what is the height here? The height here is information content. It basically tells you the surprise level of finding this distribution of nucleotides at that position versus that distribution versus what you would have expected by chance, which is roughly 25% for each of the different nucleotides. So that is measured in bits of information. So 2 bits of information means that you have 2 answers to yes/no questions. For example, is it an A or a G? Is it an AG or a CT? Yes, no. And then based on that, is it an A or a G, or is it a C or a T? So 2 questions gives you 1 bit of information, 2 bits of information, and that gives you 1 nucleotide, 1 out of 4 options. And then this is just 1 yes/no question because it only gives you 1 bit of information, so on and so forth. So that's where these motif logos come from. And there's some limitations to these models. Number one, they assume independence of the different positions. So if I have an A here, it doesn't tell me anything about whether I should have a C here or a T here. But you could imagine some cases where every time I see an A, I also see a C here, and every time I see a G, I also see a T here. And you can represent those by combinations of PWMs. Basically, the PWM itself doesn't allow you to define interdependencies, but you can have a tree of PWMs that would allow you to do that. The second assumption is fixed spacing, which basically means that if this transcription here binds, for example, as a homodimer, and it can recognize one part of the sequence and another part of the sequence, which are actually usually reverse palindromes. So that basically means that if I read this sequence from one strand, it should match that sequence from the other strand because of the way that the transcription factor is binding. In some cases, you might actually have this homodimer or heterodimer bind at varying spacings, And that would allow you to bring this part of the motif closer to that part of the motif with some flexibility. That's also not captured by this model. Why am I telling you this? Because there's plenty of room for improvement, because you could devise for your final projects or your research projects a lot of additional compute power to go beyond where the field is now. Why do we care about all these motifs? The reason is that they are the language of gene regulation, and also when there are disease-associated mutations, most of the time, 93% of the time, these common variants that are underlying most complex disorders out there are in fact disrupting not the protein genes, not the protein coding sequence, but instead one of those gene regulatory motifs. Okay, so in 93% of cases, the genetic variants associated with disease do not disrupt the proteins directly. Instead, they disrupt the gene regulatory machinery. Why is that important? Well, in our own work, we basically looked at one such example of a non-coding association with obesity, the strongest genetic association with obesity, which sits here in the first intron of this FTO gene, which stands for fat and obesity associated, which has 89 common variants, none of which perturbs a protein. In fact, you can see the tiny little exons of that gene. There's exon 2, this tiny little snippet over there, and exon 3, which is slightly bigger, but still tiny compared to the super, super long introns. So that basically means that we have no idea how these regulatory variants and which of these regulatory variants are actually important for gene regulation. That's where regulatory genomics comes in. And basically by studying comparative genomics, regulatory motif enrichments, etc., we are able to pinpoint that in fact, out of all of these nucleotides, it is specifically this single nucleotide polymorphism which disrupts an AT-rich motif by changing this T into a C, thereby losing the binding of this AT-rich interacting domain protein, which sits at the core of a gene regulatory circuitry that defines the downstream fate of your fat cells as they develop. So your pre-adipocytes in the first 3 days of differentiation decide whether they're going to be a fat burning cell or a fat storing cell. And what we found is that a single motif alteration, one of those little things that we're going to be talking about today, is in fact responsible for loss of binding of this ARID5B receptor in this massive 12 kb super enhancer, which then leads to de-repression of IRX3 and RX5, which are master controllers of this process, leading to a shift from burning calories in your brown adipocytes to storing calories in your white adipocytes. So this is extremely important for regulatory genomics of disease. 93% of cases of genetic variants associated with disease through these complex traits and these genome-wide association studies are in fact pinpointing non-coding regions, and the methods we're going to talk about today are paramount for discovering these motifs and understanding these roles. So there's a lot of technology for probing gene regulation. So we talked about how DNA is wrapped around nucleosomes, how nucleosomes are packaged up to fit 2 meters worth of DNA inside each of your cells. And now we have technologies for figuring out where are these different transcription factors binding. So on the experimental side, we basically have chromatin immunoprecipitation and histone immunoprecipitation. So we can basically pull down either a transcription factor that binds DNA to figure out all of the genomic locations where, say, the CTCF binding site binds, or transcription factor binds, or the, any of the transcription factors are binding. So you can pull down these transcription factors, chop up the DNA, and then sequence the part that came down with the transcription factor to find all of the locations where it's bound. Similarly, you can actually build antibodies against these histone modifications. So when we say, oh, this is an enhancer region, you can actually build an antibody that will pull down all of the regions that have that enhancer mark. And then you sequence the heck out of this, and you end up with this kind of profile. And this profile basically tells you where was this histone modification across the genome, and where was that histone modification across the genome. So what you end up with is regions of regulatory importance, where you can say, aha, this is an enhancer mark, so I know where my enhancers are. This is a promoter mark, so I can know where my promoters are, and so on and so forth. This is not the only assay. You can use chromatin immunoprecipitation to pull down specific modifications, specific colors of chromatin, but you can also use this assay for transposase accessible chromatin, or ATAC-seq, to pull down all of the regions that these transposases can cut. So you can basically throw transposases in the genome, figure out where did they bind, and based on where they bound, you can have an inference that that region was accessible. So this tells you about specific histone modifications or specific transcription factors. This just simply tells you stuff happens here. There's gene regulation happening here. And you can also look at DNA accessibility through DNase, which directly cuts DNA. And then that gives you very different cutting profiles associated with different regulators. For example, CTCF shows this, pattern here, which is reminiscent of nucleosomes. These are the beads on a string that position the histone marks surrounding the DNA, and so on and so forth. Okay. The last technology you should know about is this 3C or 4C or 5C or high C, which is chromatin conformation capture, or carbon copy chromatin conformation capture for 5C, or high C. We basically couldn't think of any more acronyms, and it's just a bunch of C's, and you wouldn't want any more C's. So high C. So what is this telling us? This is basically telling you which regions are looping around to what other regions of the DNA. So the way that you can envision this is taking a noodle soup, chopping up your noodles, and then religating noodles that were proximal to each other. And when you find a chimeric noodle, namely something that doesn't quite match the endogenous sequence, you don't simply religate the red to the red and the green to the green, but instead you ligate the green to the red. You can basically know that, in fact, these two regions were hanging out with each other in the soup of noodles that is the DNA inside your nuclei. So you can mark these chimeric locations by using a biotin label and then pulling them down and then sequencing them systematically to basically figure out who is interacting with whom. And you end up with these types of profiles that are basically telling you what is the folding pattern of every single location in the genome by pulling down these chimeric regions. All right, so that basically gives you a sense of what are the looping patterns in your DNA and what are your topologically associated domains. So this gives you a hierarchical, in many cases, view of the genome, where these long distance regions are interacting with each other, separated by these segments that separate the binding. And very often these segments were found to, in fact, be bound by the CTCF regulator, thinking that, in fact, the CTCF regulator is involved in establishing those patterns. So we can zoom into those, scroll across the genome, and then start getting some insights as to what the genome actually looks like. And you can see these patterns here where there's the same region interacting with many other regions, where there's, an interaction out here, another interaction there, and so on and so forth. So the CTCF locations, as I mentioned earlier, are in fact thought to be guiding the looping by this concept of loop extrusion, where you basically have the CTCF binding, and then through that, you push through the DNA, creating these loops. So you would have a binding site for CTCF there, another one there, and in between, they would sort of push out the DNA and then create these looping patterns. All right, so now let's dive into the computational methods for studying all this. So before deep learning, what did we do? We basically said, okay, great. There must be a co-regulated set of genes, must have some kind of co-binding of the same transcription factor. So you're basically doing a lot of gene expression experiments, and then you're clustering all of the genes together that show a similar expression. And you say, well, those genes are probably co-regulated. So we're going to use this to define modules of genes that are co-expressed and likely co-regulated. And then you can say, well, how do I find common regulatory motifs between them? I could align all the promoters to each other. I could use some expert models for what motifs should look like and then look for those. We could find the median string between them by enumeration. We could start with conserved blocks in the upstream region across evolutionary conservation by looking at comparative genomics, because regulatory motifs are more usually conserved. And once we have all these priors, if you wish, you can then say, if I knew what this motif looks like, I could search it in every one of these regions. Conversely, if I knew where the motifs were, I could align them on top of each other, like we saw before, and create a position-specific matrix for finding them. But if I know neither of the two, what I'm going to do is an iterative learning approach, which is going to simply search for common patterns that are recurrently found by randomly initializing all kinds of places and then letting that initialization climb up some kind of optimality criterion to find those motifs. So for example, I could start with a random sequence pattern and then search for that pattern, find where it aligns, where it matches the best, and then refine the motif definition to better match all of the starting positions. How is that even supposed to work? It's supposed to work because any slight deviation from 0.25 expected background distribution will lead to this random motif that you started with, perhaps having more of a G in that location and more of a C in that location and more of a C in that other location, will sort of veer towards those locations that start with those C's and G's and T's, and then find additional matches that are reaffirming that random initialization, and then eventually climbing up to a local optimum of motif occurrences that are common among all of these now aligned, say, promoter regions. Okay, so let's see who's with me so far. Please give me a thumbs up if you guys are following. Wonderful. So having now established this, iterative procedure, I can basically run it in a more formal framework, for example, expectation maximization. So I can make sure that probabilistically I'm learning a latent set of variables, and those latent set of variables are either the motif definitions or the motif instance starting locations. So the key idea is that I'm learning through this application and this filtering through the real data, every single time pushing my motif definition to be a little closer to what the true data actually looks like. So the key idea is this iterative procedure for estimating both the motif starting positions and the motif matrix, given some uncertainty. So it's a learning problem with hidden variables. And those hidden variables you can think of as either the starting positions or the motif occurrences. And there's many experimental methods for doing this as well. And there's many ways of doing this beyond expectation maximization. For example, Gibbs sampling is another approach very similar to expectation maximization, only instead of integrating over all of the occurrences of the motif, it basically says, I'm just going to sample from the posterior probability distribution of every starting position. So you can imagine every position, every index position of your sequence here, having some probability of starting. And it's saying, okay, if this has 10 times a score of 10, and this has a score of 3, and this has a score of 2, I will pick the 10 3 times more frequently, but 1/3 of the time I might pick the 3 because it actually has some non-random probability. So I will be sampling from that. So that's basically Gibbs sampling, where you're iteratively sampling these positions, and then you're rebuilding the motif profile from those. Another way to do that is enumeration with some wildcards. You basically allow for global enrichment, some background score, and then do that. And there are other methods that try to look more specifically at the peaks of TF binding. So basically, when you have a higher peak here and a lower peak here, you don't just simply say, oh, this is a peak and that's another peak. You say that's a 10 times stronger peak and that's a 10 times weaker peak. So I'm going to use the correlation with the peak intensity for choosing my motifs. There are other methods for genome-wide discovery of motifs. So in my own PhD, for example, I developed a method for conservation-based discovery, where across the entire yeast genome or across the entire human genome or fly genome, we were able to build these motif conservation scores that allow us to score every motif instance based on how frequently are all of these instances conserved. And that allowed you to now start discovering motifs globally without necessarily having a set of co-regulated genes in advance. And that we defined many such scores. One was a genome-wide score. Another one was whether it's biased to be upstream or downstream of the transcription start site, or in the 3' UTR versus in the promoter region, and so on and so forth. And there's also in vitro methods for defining those motifs. So now let's dive into how do we actually use deep learning for this discovery? How do we use these super cool convolutional neural networks that you've learned about to discover regulatory motifs de novo? So here's the key idea. With convolutional neural networks, what you're actually learning is hidden representations of your underlying data at multiple scales of resolution. For example, with image analysis, you might be seeing a lot of filters, a lot of kernels, a lot of convolutional filters that are reminiscent of an edge detector. So you can basically detect edges, and then you can have edges and corners at the lowest level, and then you can have these edges coming together into basic shapes like squares and triangles in one level up. And you can have those basic shapes coming together in other primitives like the eyes when you're doing face recognition, or the nose or the ears, or the wheels of a car when you're doing recognition of objects in cities and stuff like that. So you can basically do this representation learning through these convolutional layers. And the beauty of convolutional neural networks is that you share parameters, that the same convolutional filter that's detecting an edge in this location of my image is also detecting an edge in this very, very far away location of an image, that I'm sharing parameters across my entire image. What deep convolutional neural networks are doing for genomics is exactly the same thing. They're basically saying, I'm going to learn hidden representations that are shared across my genome. And those hidden representations are in fact learning the language of DNA directly. One very simple way to encode the DNA information is using this one-hot encoding. So you have one vector for T that only lights up when you have a T. You have another vector for G that only lights up when you have a G, and so on and so on forth for C and A. So this one-hot encoding is the same way that you can have image recognition with RGB channels, for example. So you have the red channel, the green channel, and the blue channel, and that gives you information about the color of every pixel in the image. In this same way, you can have 4 different channels, each giving information about the letter of that DNA, and together spelling out the actual sequence of DNA. So you can now learn your convolutional filters, which actually correspond to the units of DNA speak that are recurrently found throughout your DNA sequence. And you're learning these units de novo. So what are we actually doing here? We're basically saying, can we find a set of convolutional filters that are very well suited for predicting whether a transcription factor is bound or not? And you're doing this operation for every transcription factor in some cases in a multitask learning scenario. What is a multitask useful for? For example, you might have 300 experiments for 300 different transcription factors, each of which gives you a genome-wide binding, but the motifs that are defining this binding are very often shared, because 2 different transcription factors might bind the same motif, or they might collaborate with the same cofactor, and so on and so forth. So it actually makes sense to search for a common underlying set of latent representations. And these are the low-level representations, which include the position weight matrices, or these intermediate level representations that might include combinations of motifs that together form a grammar, or combinations of parts of a longer motif. For example, the CTCF binding site might be learned in 3 parts, one for each 3 cores, and combined into a full binding site in a higher level of representation. So you can now learn those intermediate building blocks, which might be the sequence patterns recognized by every domain of a protein that binds DNA. And then at the second level, you might have every combination of domain found in the set of transcription factors that you've done experiments for. Okay, so let's do a quick poll of who's following me so far. So give your thumbs up, and maybe you guys can actually type your questions as I will be right back.
Does anyone have any questions for Manolis here or something?
I am back. So if you have any
He's back,
questions, I'm
so
happy to take them.
go to it, Manolis.
Okay, awesome. So this is basically the basic architecture. So if you guys get this, you are all set. Everything else I say is almost superfluous. So if you're with me on this one, you've got it. Okay, so now I'm going to add layers of complexity, but the basic idea is we're going to have a prediction task. And in deep learning, the prediction task is very often even an excuse. Like your prediction task could be predicting the left side of the image or a missing square from your image from the rest of the image. That's a bogus task. You don't really care about the answer of that task. And similarly, in understanding the language of DNA, you might actually not care about the binding of all of these different transcription factors. All you might care about is whether disrupting this motif will have an impact on disease. But by creating these tasks, by creating these challenges that your system has to solve, that your learning system has to solve, you're forcing it to learn a representation of your sequence and effectively to learn the language of DNA. So now let's talk a little bit about what are these potential tasks. One task might be predict the binding of 300 transcription factors from the ENCODE project. Another task might be predict the DNA accessible sites of 100 different cell types from the ENCODE project. Okay, so carrying out systematic experiments and then tricking your deep learning system to predict the output of these experiments forces it to learn a set of representations for your for the language of DNA. Okay, and those representations can allow you now to do almost any task. So you might be binding you might be predicting whether something is bound or not, how strongly it is bound, where the nucleosomes are positioned, where DNA accessible sites are in a particular cell type, where they are across many cell types, whether something is shaped one way or another way, and so on and so forth. So the world is your oyster. You can basically truly mix and match the prediction tasks. And then let's talk a little bit about the architecture. So very often we basically have these convolutional filters, these kernels, which are capturing individual position specific matrices or position weight matrices. So what are these doing? These are basically capturing regulatory motifs. So you might want these matrices to be on the length scale of a typical motif, or you might want them to be on the length scale of a typical binding site for a single protein domain, and then sort of build up these motifs layer on, higher up. So these are the convolutional layers, which are basically telling you, how do I combine information from multiple neighboring positions to score whether that representation, that building block, that edge, or that wheel, or that nose is found in that part of the image. So I'm scanning my convolutional filter, which might look like this: G, C, A or G, G. So this is a convolutional filter, which I can now scan across my image. And if I scan this G, C, A or G, G motif here in this ATGC sequence pattern, it will not give me a very high score. But if I scan the same motif a little further, so I've moved now by 2 nucleotides further, and the sequence is GCAG, suddenly it matches GCAG. And that actually has a high score of that kernel, of that intermediate feature that is learned from your DNA sequence actually occurring at that location. Okay, so give me a thumbs up if you guys are with me on how I'm actually scoring these convolutional filters as I'm scanning them through my sequence. So awesome. I see a bunch of thumbs up. Cool. So you guys should either type your questions or unmute and sort of ask your questions if anything is not clear. So we are now able to scan these convolutional filters, and we're able to learn these convolutional filters through the sharing of parameters to learn a set of primitives that are recurrently found across the DNA. So these are the convolutional layers. And then at the higher level, I'm learning combinations of these motifs and so on and so forth. And then I want to know, did a motif occur at that location or not? And that's where the pooling layers come in. You learn about pooling layers. You can take the max or the sum or some other function over a region of DNA. Why? Because frankly, I don't care exactly where the transcription factor bound. It might bind a little more to the left, a little more to the right, and so on and so forth. And therefore, it's very important to be able to aggregate information across many, many different starting positions to say that indeed there's a match that could have led to a binding event in that location. And that's what the max pooling layers are doing. So I'm going to ask for one more thumb up to basically see if you now see all of these different layers of architecture. At the bottom, we have this one-hot encoding. Right above it, we have these convolutional filters that can be multi-layered, learning basic primitives at the bottom and then higher order primitives at the top. And then we have this max layer, which is basically teaching us about whether these motifs occurred there or not. So what is the system doing when it's learning across this whole thing? It has a predictive task, and it's learning representations whose occurrences are relevant for that task. Okay? And then we basically have these traditional non-representation based multi-layer, fully connected component of our network that is basically very useful for putting together all these representations that have been learned for your ultimate predictive task. So here's where it becomes very cool. You can basically pre-train all your model using hundreds of transcription factor bindings, patterns from ENCODE, and then keep the learned representations, keep the bottom of your network, and then switch up the top and basically say, I'm going to throw the fully connected layer. I'm only going to keep the learned representations and now try to predict all of the accessible binding sites for hundreds of cell types. Why is that, even a thing? Because the same regulatory motifs are responsible for the underlying code of the DNA, the underlying language of DNA, and you can learn that code using any task and then switch around the set of learned representations by a different fully connected network for another task at hand. For example, you might learn about binding of factors and then turn around and try to predict SNPs that are important. You can now say, well, my prediction task, my loss function guiding the training of the network is now going to come from whether a motif was found to indeed perturb binding of, where the perturbation of the motif was indeed found to perturb binding of a transcription factor or enhance reactivity and so on and so forth. So you can switch this around and create new predictive tasks from your previous architectures. And that's a form of transfer learning, okay? So we talked about convolution, extracting information, these convolutional filters, the filtered signal, and then your denoising, your max pooling. And then we talked about this ReLU layer that you guys have learned about, how you can use that to say, well, I don't care for negative occurrences of the motif. I only care about positive occurrences for the motif. So you can use this rectification linear unit to say anything above a particular threshold, I'm going to even pay attention to. Okay, so you can put all of that together with your convolutional module that learns representations, and ultimately your prediction module that learns their combinations. And what's really remarkable is that when you apply these convolutional neural networks to deep learning data, to regulatory genomics data through deep learning, what the deep learning methods are doing are in fact learning representations that match your position weight matrices and your position specific scoring matrices. Okay, so you're able to learn these representations systematically. Okay, so I'm going to skip some of those slides.
Manolis, there's a question for you in the chat.
Oh, okay. Sorry. Let me pull it up. Can you say again how you chose the kernel's filters, how you chose the order of base pairs, and which ones that are in your kernel filter? So this is done through my prediction task. If I'm trying to predict where the transcription factor is binding, some filters will be completely irrelevant to that. So what I'm going to be doing is back propagating through this and then saying which, so if I have a loss function, I'm predicting across the genome what are all of the binding locations, and I get a bunch of these locations wrong the first time around. So I'm getting error from these wrong locations that I'm predicting incorrectly, that's feeding back through my loss function, through this gradient learning and this stochastic gradient descent that you guys have heard about. I can now propagate back the error from all of the incorrect predictions that I'm making, and then update, of course, my fully connected layer, but also the representations. And those representations that are more relevant to the predictive task at hand will be favored and upweighted and refined to match the motifs of my of the actual motifs that matter, of the protein domains and the regulatory motifs for the different transcription factors that are actually relevant to the predictive task at hand. Did I answer your question, Sarah? Awesome. Okay, this is a great question. Are there any other questions? Okay. Type them into the chat and they will be answered. Okay, so we basically have the ability to now learn all of these weights through this back propagation to actually start learning these internal representations. So we basically start with a one-hot encoding at the bottom of our DNA sequence. We then learn these convolutional filters. We then use the thresholding to say, did that motif occur in any one location or did it not occur? I don't care about negative scores. And then your max pooling layers that are telling you about what, 1 second. Dalu, I'm really sorry I can't answer right now, but yes, please do come. And then that allows us to figure out if the motif was occurring in that, say, 50 nucleotide window or 500 nucleotide window and so on and so forth, then feeding back into your fully connected layers at the top, okay? And you can have multitask learning, which is highly recommended in these cases because it forces your representations to be generally helpful, to be very widely applicable. All right, so there's been a lot of methods for doing this. So the first deep learning method for regulatory genomics was DeepBind, that basically used a lot of these these protein binding microarrays these selects experiments that are sort of iteratively searching for regions that are bound by a set of transcription factors, these ChIP-seq and CLIP-seq experiments that are looking at DNA and RNA binding, and built all these large-scale data sets and then put them into this deep learning framework through a graphical processing unit to basically learn a set of motifs that then can be combined to predict gene regulation, to predict the impact of regulatory motif mutations in disease, and to also detect binding sites. And at the bottom of it, you basically have these convolutional layers that are motif detectors. You learn these motifs, you have the ReLU and the pooling layers that basically threshold to say, did a motif occur somewhere? And then a fully connected neural network that allows you to predict this. And then, of course, the prediction error is back propagated. And then you update all of the above to continue iterating and learning. Okay. And this actually did fairly well at the time, and has been, of course, greatly surpassed by many next generation models, but it at the time surpassed many of the previous models. And what was really kind of cool is that the both DeepMind and other methods that followed were in fact able to recapitulate the learned motifs. So this was a motif that was previously known for PRDM1, and then DeepMind from either selects experiments or from chip experiments learned motifs that are in fact very close matches to what was previously known. And that's very satisfying, the fact that your convolutional filters are not just a black box that you're never looking at, but if you look at the parameters of your convolutional filters, they're in fact matching known motifs, which is, again, very reassuring. So, and then of course, this is very exciting because you can then turn around and say, what is the impact of a disease mutation? So you could say, if I change this one letter G into a C, how much will that impact my overall prediction? And when you do that, you basically see that the places that are predicted to have the strongest impact on the gene regulatory scores are indeed the places where motifs are occurring and where motifs are known to be bound. And that makes a lot of sense. It's almost just making sure that the model is doing what you predict, what you trained it to do in the first place, namely recognize regulatory motifs, because in those places where no motif is occurring, a mutation will not change your model at all, because the convolutional filters that you've learned are simply not detecting these locations are as relevant at all. DeepC was a subsequent model that was trained directly for predicting the impact of regulatory mutations using this impact score to train directly on GWAS and EQTL, so genome-wide association studies, and expression quantitative trait loci that you guys have heard about before. And then Bassett by David Kelly and John Wren was basically again, learning this concept. Now it's flipped from the bottom going up towards the bottom of the page. And then again, learning these motifs and, matching them very nicely. Okay. So that's the foundations of convolutional neural networks for regulatory genomics. You can then start doing this multitask learning that I alluded to earlier. And you can start asking, how much is every neuron in my deep neural network firing for different classes of elements, and what are the shapes of those firings, and how well am I predicting those, and so on and so forth. So you can incorporate high-C information in your systems to basically say, not only do I have the linear binding of particular transcription factors, but I also have information about their interactions and how they're looping in 3 dimensions. And you can feed that through your convolutional neural network as additional layers of information, additional features for your prediction. And you can change the architecture to specifically do that. As I mentioned earlier, these motifs are not just found in enhancer regions and in promoter regions, but they're also found in splicing and in many other locations. So there's been a lot of work in predicting tissue-specific splicing and the tissue-specific splicing code by changing the training to instead of saying what's bound and what's not bound at each location, to say, is an exon excluded or not in a particular cell type? And then you have an inclusion score, an exclusion score, and so on and so forth, which allows you to start asking, how well can I predict whether that exon will be included or excluding using a convolutional neural network that's looking at all of these neighboring sites. For example, the 300 nucleotides flanking the exon of interest, the 300 nucleotides flanking the exons surrounding my exon, and the exon itself for both the one that I'm trying to predict and for the ones before and after it. And using this through hundreds, actually thousands of exons and thousands of RNA features, what Xiong et al. And many others were able to show is that you can indeed start predicting splicing. And this actually tells you how much you're predicting the impact of these mutations to act on splicing. And what you can see is that the exon itself has an enormous amount of information encoded about splicing, and the introns have also an enormous amount, but mostly proximal to the exons themselves. So basically, as the RNA splicing machinery walks through the intron, it reads a lot of information. It's also reading a lot of information from the exons, and it's sort of making the decision to include something or not. And we can read now the language of that RNA processing machinery to start predicting the impact of intronic variation, exonic variation on splicing, and start predicting splicing itself. I had also prepared some slides on single cell RNA sequencing, but I understand that you might have some other courses to go to. So, there's been a lot of cool technologies for RNA sequencing, and you can use that for probing RNA and DNA accessibility. And then you can use all of these single cell data sets to start predicting the trajectory of cells across different states, to start predicting the dynamics of which genes go up or down in expression, to start predicting different cell types, to basically distinguish the universe, if you wish, and to also start asking about what genes are differentially expressed, what pathways are differentially expressed between different conditions, and so on and so forth. And then deep learning has a huge impact in single cell RNA-seq analysis as well, because it allows you to learn latent representations from the RNA-seq data now, rather than the DNA accessibility or the TF binding data, that can then be informative in any prediction task that you would like. For example, is my individual healthy or sick? Do they have Alzheimer's or not? And then you can create these deep learning tasks with the underlying super high dimensional representation of 20,000 genes and their expression levels to learn lower dimensionality representations that are relevant for the prediction task at hand. And there's been a lot of deep learning methods for single cell RNA-seq for basically learning these autoencoders. For example, you can say, can I predict the input data set from a lower dimensionality representation? Can I create those deep embeddings that can be used to predict my input data set, and therefore learn a representation that's relevant? And why is that useful? Because, for example, if I have RNA data and DNA accessibility data, I might be able to combine them by building translators between the two that translate them through these latent representations. And there's a lot of work in doing this. Another very cool method for single cell analysis is this single cell variational inference that is actually using a neural network to estimate the parameters of variational inference models that allow you to, again, predict the cell types, predict the contribution of noise, predict the contribution of different cell states, of different pathological conditions, and so on and so forth. And you can use a lot of information through these bottleneck layers to actually start learning these representations. Okay, so and again, you can use that for all kinds of studies to basically look across different studies, across different organs, across different species, across different cell states, and then learning these representations. And a very cool, powerful idea is that in your encoder itself, you could include additional state that is basically telling you perhaps about the labels of your different studies. And you could force your network to use these labels to predict the output, or you could force your network to be independent of these labels to learn a label independent representation. And you can use fixed labels, you can use trained labels, you can do different queries using these labels that allow you to both learn representations as well as context dependent modulations of these representations based on the task at hand. So you can use all this information to really train your own networks and do just an incredible wealth of different tasks. I think it's time, so I'll stop there and see if there's any remaining questions. If not, wish you guys goodbye.
Oh, that was a great job, Manolis. Any questions? Well, I think we're good. I think it was a bit long, but it's very good. It was a great thing, Manolis. We'll get your slides and put them up, and we really appreciate that very much.
Sounds great. So you guys, if you want to download the slides, they're from this link that I pasted at the beginning.
Yep,
All right.
we got it.
Thank you so much, Mark.
Thank you, Manolis.
Thanks for the opportunity. Bye, everyone.
Take care. You take
Bye.
care. Bye.